discuss about ventricular system of the brain. What is the role of ventricular system?
Produce CSF.
Produce CSF and also help in drainage of the CSF in subarachnoid space in central canal of the spinal cord also. So how many ventricles you have got?
Four.
Four ventricles. Four ventricles. This view is basically your sagittal view. Here is your corpus callosum, here you can see. But this is your lateral ventricle. Lateral ventricle, you have got two of the lateral ventricles. And lateral ventricles are in your cerebral hemispheres. Third ventricle is in your diencephalon, which is also a part of the prosencephalon of the forebrain. So lateral ventricles, they are connected to third ventricle by this special foramen, and this foramen is also interventricular foramen, or this is also called as foramen Monro. So it means you have got two lateral ventricles. How many? Two lateral ventricles and one third ventricle. Now look, this third ventricle, third ventricle. This third ventricle is basically a space between two thalami. Here you can see that, this is also a sagittal view, yes? Here you can see this gray structure is basically your third ventricle and this is your thalamus. So it's mean on basically either side of third ventricle you have got thalamus. So this will be right thalamus, this is left thalamus. In between you have got this special cavity, that is third ventricle. And this third ventricle is receiving CSF from these lateral ventricles through this foramen Monro. Foramen Monro. So two foramen Monros. How many foramen Monro? Two foramen Monros. Or this is also called as interventricular foramen. They are two in number. You understood? Yes, and then look on this reddish structure. This is very important structure. Here you can see also this fourth ventricle. This is called as choroid plexus. This is choroid plexus. Choroid plexus of ependymal cells. Which cells? Ependymal cells. And this is very important because it helps in production of the entire CSF. So it means that CSF is produced within these lateral ventricles or third ventricle or fourth ventricle only by these special cells and choroid plexus of the ependymal cells. And CSF is basically flowing in this complete ventricular system or subarachnoid space by special epithelium, which is ciliated columnar epithelium, which is lining this entire ventricular system or the central canal of the spinal cord. You understood?
Yes, sir.
So it's mean you have got two lateral ventricles, one third ventricle, and then this third ventricle is communicating with fourth ventricle by this narrow duct, and this duct is present in your midbrain, and this duct is called as duct of Sylvius. Duct of Sylvius. And duct of Sylvius is basically connecting your third ventricle to the fourth ventricle. So you have got lateral ventricles two, one third ventricle, one aqueduct of Sylvius, which is connecting your third ventricle to the fourth ventricle. So total you have got four ventricles, two lateral, one third and one fourth ventricle. Now basically you know that from fourth ventricle now CSF is flowing into the central canal and spinal cord and in subarachnoid spaces. So in fourth ventricle you have got three holes. Three holes are foramens or apertures. Aperture, this is also a hole. So two on the lateral side, two hole on the lateral side, those are called as foramen luschka. And they are draining basically CSF into subarachnoid space. Which foramens? Two foramens luschka. And one on the medial side. One on the medial, that is foramen Magendii or Magendii, okay? Yes. So remember lateral, luschka and medial side. So two, you have got foramen luschka on the lateral side and one foramen on the medial side. That is foramen Magendii. You understood? Now third thing, which is very important, that role of CSF, that this CSF is basically produced daily up to 500 cc. Up to 500 cc or up to 500 ml. But space or the capacity of this entire ventricular system or this subarachnoid space, here you can see also in this structure that this is your basically you can see that this is aqueduct of Sylvius, this is fourth ventricle, here is your cerebellum, and it's behind pons in front of cerebellum. So this is basically roof of fourth ventricle formed by your cerebellum. Here you can also see. Yes? And then this is aqueduct of Sylvius. This is your third ventricle, lateral ventricles, interventricular foramen also. So what happens that basically, this CSF is produced daily up to 500 cc or 500. But the capacity of this ventricular system or subarachnoid space which is draining this CSF is only up to 90 to 150 ml. You understood? Yes. So rest of CSF is basically, absorbed by arachnoid cells, arachnoid villi or arachnoid granulations. Arachnoid villi or arachnoid granulations. So it means CSF up to 350 up to 500 ml. Now CSF is basically produced or absorbed by these special cells, and these are called as arachnoid villi. And it's moving or flowing within these channels by special lining of these tract with the help of the ciliated columnar epithelium. You understood?
Yes, sir.
And there are special movement pulsations within these ventricular system which are they are helping in pushing or the movement of this entire CSF within these basically channels. So clinically what is significant that function of these CSF, it's provide nutrition to your CNS, act as cushion, yes, prevent your CNS from the infections, and it's present basically in two layers of meninges, arachnoid matter and then pia matter, that is subarachnoid space. Within the arachnoid and pia matter, that is subarachnoid space. So what happens that if suppose there will be blockage of this tract, whether the blockage in this foramen Monroe or in this aqueduct of Sylvius, this CSF is now accumulating in these lateral ventricles. Size of the lateral ventricles will be increased. That is called as hydrocephalus. That is called as hydrocephalus. So simply you have to understand that hydrocephalus. What is hydrocephalus? Hydrocephalus, it means accumulation of CSF accumulation within ventricles and increase in pressure of CSF. So it will increase the size of the ventricles, lateral ventricles. You are increasing the size, it means you are compressing these all upper structures of the brain. So there will be atrophy, decrease in size of the brain. Decrease in size of the brain, it means mental retardation, decrease IQ level. Second thing, you are compressing all the structures and you are pushing lot of pressure, putting lot of pressure on these within these meningeal spaces also. So you are compressing the special structures and compressing the optic nerve tract also. So you will have problem with the vision like diplopia. You will have swelling of the optic disc like papillary edema in case of the hydrocephalus. You understood? You will have headache. So blockage is common in this part, narrow part, that is aqueduct of Sylvius, or in this interventricular foramen. Or what happens, like we have discussed, that anomalies, one is cherry 1 and cherry 2, especially cherry 2 malformation, that when there is displacement of the cerebellar vermis and tonsils through this foramen magnum, and that is also blocking this tract. So it means that blockage of any structure, whether this is embryonic defect or in later part of the life you are having blockage of this part, maybe due to the tumor, maybe due to the bleeding in subarachnoid space. So that it means that is basically non-communicating hydrocephalus. It means obstructive or the non-communicating. Obstructive or non-communicating hydrocephalus. So obstructive or non-communicating hydrocephalus is basically problem within the tracts or within the structure. So cherry type 2 malformation, because it's also blocking due to the displacement of the cerebellar vermis and tract, that is also associated with which type of the hydrocephalus? Obstructive or non-communicating. You understood?
Yes, sir.
Third, you have to understand that what is communicating hydrocephalus. Communicating means there is still a communication within these channels, but you have got problem in arachnoid villi, arachnoid granulations, that there is problem in the absorption basically. So CSF is produced, but now there is lack of the absorption. So lack of absorption, it means there is that type of hydrocephalus is called as communicating hydrocephalus. So in case of the hydrocephalus, when you will witness that increase in size of the ventricles, you are performing some procedures like shunting. You will insert the shunt. Yes. Yes, ventriculoperitoneal shunt. Later I will show you the structures also. So you have to understand all these special tracts, how many foramines there, how many foramines in fourth ventricle, what is role of this foramen, how it's produced, which help in production, and what are the special cells which help in transportation or the absorption of CSF. This is clinically very important. So here you can see, this is what we have discussed. That you can see, this is basically your lateral ventricle, yes, and this is choroid plexus. And then CSF is coming to your third ventricle through this foramen Monroe, and then through this aqueduct of Cilius. Yes, this is your fourth ventricle, and this is your lateral aperture or the lateral hole foramen luscus, and this is choroid plexus of the fourth ventricle, and then it's flowing basically in subarachnoid spaces, and then through these arachnoid granulations it enters to the sinuses like superior sagittal sinus. And then it's flowing in this subarachnoid space and central canal. It's been spinal cord is also floating, in this subarachnoid space in CSF. So that's why I have told you that spinal cord ends at the level of L1, but subarachnoid space extend up to S2. So in case of problems, relating to the CSF, whenever you have to approach to the subarachnoid space to drain CSF for investigative purposes, first, what is the landmark of this LP lumbar puncture? Between L4 and L5. So you have to understand what are the structure you are going to, basically puncture or pierce to approach to subarachnoid space. So landmark is best, is the safest is L4 and L5. So first, you are approaching to skin, then after skin subcutaneous tissue. Subcutaneous tissue or subcutaneous fat, and then you have got ligament on the posterior part of backbone, the supraspinous ligament. Which ligament? Supraspinous. This is basically pathway of your needle while performing this procedure. Supraspinous ligament, and then interspinous ligament. Which ligament? Interspinous ligament. And the ligament which in within this basically bony channels of the vertebral column, that is ligament flava. That is a ligament flava. This is clinically very relevant ligament, because if there is any increase in size of the ligament flava, it lead to decrease in size of that special foramen through which the spinal cord pass. So hypertrophy of this ligament flava, it means it's lead to basically compression of the spinal cord. So third structure is ligament flavum. After ligament flavum, now you have got epidural space. Epidural means above the dura, and dura is the first layer of the meninges. Yes. So epidural space. Epidural space. Behind epidural space you have got dura, and behind dura you have got arachnoid. And then you are approaching to subarachnoid space, and there is your CSF. So 1, 2, 3, 4, 5, 6, 7, 8, 9. You have to remember this same sequence. You will have questions relating to this also in your exam. So you understood? Yes. And then another structure. Look, this is the coronal section. In my first part of the this ventricular system lecture, I have explained, I have drawn this structure also, how CSF is flowing and what will be the coronal view. The view we have discussed, that is sagittal. Yes, this is the, here you can see, these are the lateral ventricles, yes? Lateral ventricles. And it's through the foramen Monroe, this is foramen Monroe coming into your third ventricle. This is aqueduct of Cilius in your midbrain to your fourth ventricle, and then through fourth ventricle through the central canal and subarachnoid spaces. On the lateral side you have got foramen muscular, two of them. On the medial side you have got foramen media, and then it's flowing to your entire basically subarachnoid spaces. Now any bleeding in the subarachnoid space, subarachnoid hemorrhage, it means worst headache of the life. due to the rupture of the aneurysms. Arteries of the circle are less. So it means the color of CSF, normally CSF is colorless. Normally it's colorless. But in case of bleeding, that when you are performing the spinal tap and you see that color of CSF is yellow or bloody, it means bleeding is subarachnoid space. Bloody CSF. Bloody CSF. And in case of the hemorrhage in this space, it also leads to the blockage of the tracts, which is helping in drainage of CSF. That is also an example of obstructive hydrocephalus. Patient may have meningitis, headache, fever, infection, nuchal rigidity, neck stiffness. That is typical picture of the meningitis secondary to the subarachnoid space. And hydrocephalus also due to the blockage of this tract. You understood?
Yes, sir.
Yes. So here you can see another structure, that this is your subarachnoid space. This is your subarachnoid, and this is your arachnoid granulations. And the arachnoid granulations, they are draining this CSF into sinuses. And this is your which sinus? Superior sinus. Arachnoid granulation draining into superior sagittal sinus. Superior sagittal sinus. You understood?
Yes.
And this is another one of the most common procedure in case of hydrocephalus, when we are connecting shunt to lateral part of this lateral ventricles into your peritoneal cavity. This is called as VPS, ventricular peritoneal shunt. VPS. And next we have to understand significance of these sinuses. That what are the dural sinuses? They are large venous channels. They are traveling through which nerve? Dura. And draining blood from the cerebral veins. Remember, draining the blood from cerebral veins. And then receive CSF from the circulations. And empty into internal. We have discussed the sinuses, and we have already discussed the significance of the cavernous sinus also, which is the most important, largest sinus also. So you have to understand, what is basically role of CSF, how it's produced, what are the channels for the drainage of CSF, and what is the basic difference in communicating and then non-communicating, non-obstructive hydrocephalus. Okay, thank you.